Okay, so let's start. Hi everyone. Today we are talking about brain networks. So have you heard anything about brain networks?
From what you said, no.
Okay. So the first part will be connected. So first we are going to talk about history, but that's not a history itself. I don't mean some ancient guys from Egypt and Ramon and Cajal and so on. I mean that the large-scale brain networks, they're quite young. So they were discovered in 2011. But before that, we knew a lot of different facts about the brain, the connections in the brain. And so, so we knew something about brain networks, and the history is our try to talk about it. So do you remember from the previous lecture, I guess, this picture? What is it about? Okay, so frankly speaking, this certain picture is not about only the evolution, but this is about a triple model of Paul MacLean, and he also decided that each part of the brain, I mean the lizard brain or brain stem, the mammal brain or limbic system, and the neocortex or human brain, this part has its own consciousness and its own aims. For example, for lizard brain, it's a survival. For mammal brain, it's connections with other with other guys from your pride. And for human brain, it's some kind of mind, some rational things. But also this this picture is connected with evolution. So the lowest parts are the most ancient, and so on and so on. Okay, so let me remind you about this lower brain. So do you remember the name of this part of the brain? Maybe this one. Yeah, brain stem. And yeah, this is brain stem and pons. And there are the most important nuclei for survival in this part of the brain. And have you seen films about killers who kill? Yeah. Oh, great. And so have you seen some killers on the top of the roof, and they're trying to shoot here? In fact, when they shot here, the bullet goes like this through the brain stem. So they're trying to kill person, because if you destroy this part of the brain, the person can't can't survive. So he can't live, he can't breathe, he can't do anything.
Like the bullet is from that side, he will not die.
You mean from this side? Maybe.
You will die with suffering.
Maybe. So also there are some films when, I'm sorry,
yeah, because I've heard of someone trying to suicide.
We cannot see the slides.
Yeah.
So and do you see the slide now? Sorry.
It's just you might have to Can you see the Zoom share? No, that's one. Yes.
Okay. So you haven't missed a lot. So we just talking about killers and some movies.
And we have them between us.
Okay, yes. Sometimes when you shot here,
Yeah.
people can survive. So they usually die because of the blood, yeah, and so on. And also, have you heard about Phineas Gage? He was a guy who walked somewhere on a railroad, and big metallic thing. He destroyed a part of his head, part of his brain, so it went through his prefrontal cortex, and he was alive.
His brain is split.
Not his brain hasn't split, but this thing just destroyed a part of prefrontal cortex, so this prefrontal part. And before this case, he was very pleasant, good person, but after that, he couldn't control himself, and he argued with people. So his character, yeah, he became. Yeah, he couldn't control himself anymore. But okay, let's continue about some other brain parts and
some other films.
Some other films, yeah. Okay, we missed something about spinal cord. So this is part of the brain, not in not in the head, but in the spinal cord, there are some very important neural circuits. I mean, pattern generator. So yeah, they generate signals for locomotion, so for walking, for running, and so on. So some rhythm patterns for motion, like for this cat. And okay, oh, I have some dates here. In 1911, so many years ago, one neuroscientist, Thomas Graham Brown, he did some experiments, and after these experiments, we knew that locomotion is not a reflex. So before his experiments, people thought that, for example, cats or dogs, they walk because they touch the earth, and it's a kind of reflex. So they
like light.
Yeah, like light. And he cut it the spinal cord, so somewhere here in the neck of the cat, and put it onto some moving ground, and cat could walk. Yeah, and that was his experiment. And after that, you're not happy about this cat. You loved movies about killers much more than these facts about the cat. Okay, and then neuroscientists found that these patterns of locomotion, they generated by special pattern generators. They look like that. So there is a flexor pool of neurons and extensor pool of neurons. So in this circle, there are many neurons, and they're connected. Mostly they're connected with another pool of neurons, and in fact they suppress. They have inhibitory connection. As you can see on the slide, when the neurons from the one pool are excited, then neurons from another pool are inhibited. Okay, and so there is a kind of rhythm. And no, not only in the cat, but also in my spinal cord and in yours and in spinal cords of worms. But worms, they don't walk like people, they do like something like that, yeah, or snakes and so on. But there are some higher levels of motion control. For example, there are pattern generators, but higher in some red nuclei, in cerebellum, and in motor cortex. There are centers that regulate this frequency of pattern generators. So the pattern generator can work like that, but then the central nucleus said to this nuclei, do it faster and do it faster, but without the control from the central brain, we can't change the frequency. Yeah. So, and in different species, for example, for lamprey, or it's kind of worm, for cats and for human, there are different parts of such regulation. So for worms or for lampreys, there are almost no other parts of motion generators except pattern generators. Okay, and we can control our movements better than lampreys. Yeah, so it looks like something like that. So in the spinal cord, there are some pattern generators higher in red nuclei. There are centers of motor control. Basal ganglia also takes part in this control, cerebellum and cerebral cortex. So quite many structures you need for working or for running or something like that. Okay, so the next part is the limbic system. Do you remember something about the limbic system? No? Nothing? Zero? You know, these weekends I was in Perm. It's a city in Russia, and there was a conference, scientific conference for children. So children of different ages, they presented their works. Some of them were quite interesting, some of them were quite naive, and one little girl, I suppose she was 7 years old. No, she showed us the brain and talked that there is a frontal lobe, there is a parietal lobe, and occipital lobe, a temporal lobe, and cerebellum. And then I asked, and are there any other structures in the brain? And she said, no, you see, only in this part. In fact, under the cerebral cortex, there is, of course, the limbic system. And as I said some slides ago here, it's the most important part for emotions and for memory and so on. So, yeah, from Latin, limbic means border or edge. And it was introduced in 1952 by Paul MacLean again. And but before the limbic system, there was a concept of puppet circuit. Oh, have you heard about puppet circuit? No? Okay. So it was introduced in 1937 by James Puppets, and he suggested that this is a chain for connection of hypothalamus and I guess, yeah, and the limbic lobe. But so there is no huge difference between Puppet circuit and limbic lobe, but limbic system is more modern. And so these are structures which are included into this limbic system. For example, a huge part of hippocampus is here, and so for many years people thought that we need limbic system just for emotions, but then we understood that it's quite important for our memory, so we can't divide the memory and the emotion.
So someone with good memory has good emotional life, and the other way around, someone that forgets so much is emotionless.
No, I mean that these emotions are mostly connected with, for example, these memories are connected with emotions, for example, with fear, with happiness. So, do you remember the amygdala? We talked about this before. So, as you know, the amygdala is the main part for fear. But it's not about only fear, it's also about some stimuli for what you feel this fear.
Yeah, it's like the trauma when someone have an accident and smell something and then smell this something again, we feel the same fear.
Yeah, that's right. So all the first memories that you have mostly in the limbic system, and then it goes to the neocortex after you sleep, for example, you know. So, and we will talk about it more when we will talk about imagination and thinking. I think a lesson after. Okay, so the limbic system is quite huge, and now we include many other structures to the limbic system. For example, part of prefrontal cortex and singular gyrus, because we need all these structures to feel the emotions. Yeah, the next circuit is a reward system.
Mm-hmm. Yes.
So, do you know something about the reward system?
Something to do with when I reward myself. Yeah, something to do with glucose, something like this. Our happiness, like the endocrine system.
And what is the main neuromediator for this? Yeah.
Serotonin, something like that.
Yeah.
Or maybe when I put some goals to meet, and after I achieve this goal,
Yeah, you have a reward. That's right, reward in your brain. Okay. So reward system is something which we need evolutionally. So to survive, to achieve some goals, but it's not quite simple, and there are different parts in the reward system, such as tegmental, ventral tegmental area, or VTA, nucleus accumbens, prefrontal cortex, and some other nuclei. And of course, the main neuromediator or neurotransmitter is dopamine. Yeah, have you heard about this?
Just one.
So, when do you feel the most dopamine in your brain?
happy or when you take some stuff or eat something
Just
so delicious? No, when you take something.
So
When you are happy.
I'm not sure
Or
that when
maybe
you are
when
happy.
you take some drugs. I didn't say that. I said something.
Okay, so for better understanding the role of dopamine, we will talk about some experiments. But before that, I want to mention that there are some special nuclei again, ventral tegmental area, okay, and substantia nigra, which has dopaminergic neurons. And these neurons are projected to different parts of the brain, like that. And or like that. And the first, in some way, the first experiment which helped us to understand the meaning of dopamine was the experiment of James Olds and Peter Milner in 1954. And so, do you guess?
Yeah, there's some cheese,
Some cheese? No, no cheese.
something for the rat.
No, in fact, in this experiment, there was a kind of button for a rat and some electrode. You see, this is an electrode, and it was inserted into nucleus accumbens, Na, and the rat pushed the button again and again and again and again and again, and so that's all she or he needed.
That's nice.
So
Only something like that.
Same hyperactive.
yeah, so the rat didn't want to eat or to drink or to do something. So, and at the end, it was very exhausted and dead.
That
Yeah.
true. Yes. Seldom happens. That's nice.
No, because it hasn't got enough energy because of lack of the food and the water and so on.
So how long it took the rat to die?
How?
How long? Like
Oh, I don't remember. You can read the article.
Also, by the way, it's a concentration of dopamine for something that's very, very huge. It's little bit dead.
I think in this case, the rat dead just because it hasn't got enough energy. So it was very exhausted. And that's why we call some parts of the brain as the center of pleasant or something like that. But in fact, there are more than one center of pleasance, so we can stimulate many other parts of the brain, for example, VTA or something here, something here. So many points in the brain will be okay for this aim. So if you want to be happy always, so yeah, you can put the electrode somewhere here and and push the button. Yeah. But in fact, we don't know if this rat was happy or not. Maybe it just pushed the button, but it wasn't happy. Yeah, you know.
Why?
It can be. For example, you want some chocolate very much, and then you eat it, and you don't feel the pleasure. Something like that.
Why they didn't apply this on monkeys on some higher level?
to ask monkeys and people.
Not to ask monkeys, but at least monkey has some has action or something like
I think it's
maybe next time.
yeah,
You can try to have a
Not already.
it's not very good in terms of ethics.
Okay.
Poor monkeys.
Marina can be volunteer.
Why I think the smaller the animal is more ethical.
Yeah.
It's unethical to go after.
So there is a concept that for example, monkeys, they closed people, and that's why we can't do something very bad with monkeys, but we can do it with rats and with worms. So that's the science. Okay, and however, in 1997, some other scientist, his surname was Schultz, he conducted very interesting experiments with monkeys. Yeah, as you can see. And yeah, this is the scheme of this experiment. Oh, you can look it here. So the monkey had some task. For example, it should put the button when it see the light. Okay, and then after I don't know, after a minute or after some seconds, it gets some juice or some sugar. Okay.
And say it well, in correct way.
No, just just put the button and then juice.
Is this related to animal dressing?
Yeah, in some way. So it's kind of conditioned reflex. And in this experiment, they measured the level of dopamine in, I guess, in VTA, in ventral tegmental area of the monkey. And this is the scheme of the experiment. So in first case, when the monkey didn't know about the rewards after the light, the level of dopamine increased after drinking the juice. Yeah, so juice, dopamine. Okay, but then when the monkey learned that after the light it will have the juice, the level of dopamine increased when it saw the light.
Wow.
Yeah, so during the light, but when it got some juice, no dopamine occurred. Okay, and but when the monkey saw the light and it won't receive the juice, the level of dopamine decreased.
Disappointed.
Yeah, disappointed. So the authors consider that dopamine is released for reward without conditioned reflex. So when you suddenly have a reward, your dopamine increased. Then after stimulus, after which there will be a reward. So for example, you open your Facebook and you always see likes under it publications. Yeah, so but when you don't see the like, your dopamine is decreased. So something like that. Okay, and there are different dopaminergic pathways. So mesocortical.
I have questions.
Okay.
In case of applications or someone applicate to eat or to have something,
Mhm.
the level of dopamine is increased in high level, or what's the level of dopamine in this situation?
So can you give me some concrete example?
If someone will be pleasure when you eat so much, so the dopamine level in this case, in the higher level, just normal.
Like the dopamine is connected to addiction. If
Yeah.
someone is addicted to eat or eat chocolate or something, so that the dopamine level is high, or it's something else.
It's the level is not high, but you have some special circuit connected with eating, for example, if you are addicted to eating, and you have very low dopamine on anything else. For example, you are not fond of learning or moving or something else because you have very, very strong circuit. For example, there are different circuits in the brain, and this is for learning, this is for moving, and if you're happy and your life is balanced, you have the similar connection with dopamine in all these circuits. But if you're addicted to something, your dopamine system produces a lot of so all the dopamine is in one circuit. Yeah.
An example of this experiment is when mom comes home and always bring gifts. So once you see her coming from job, like you are expecting, something. It's something.
Yeah, that's right. Good example. Okay, so there are three dopamine pathways: mesocortical, mesolimbic, and nigrostriatal. And the mesocortical goes to the cortex, and mesolimbic goes to limbic system, and nigrostriatal is connected more with the movements. Okay, and also there are some other pathways, for example, serotonin pathways, norepinephrine pathways, and as I said, I think on the first class, almost all of the neurons in the brain express glutamate. If they excitatory neurons, they express GABA if they are inhibitory neurons, and dopamine, serotonin, and some other neurons, they are special, and they have some special nuclei in the brain, and they have projections to almost all the parts of the brain. And if you're close to artificial intelligence in some way, maybe you have heard about reinforcement learning. Have you heard about this thing? Yes? And have you heard about some other kinds of learning? So there are three types of learning for artificial intelligence, and what are they?
Daniel's now.
No, I know something about game theory, but I don't know.
Okay, so let's talk first about the reinforcement learning. It was made for some intellectual agents or robots or something like that. So we can do some kind of reward for a robot or for an agent. For example, I don't know, if we have got a cleaner robot in the room, we can take a reward to this robot when it has some rubbish. Okay. And so after some cycles of learning, this robot will find this rubbish by himself, just because of this process. And
Like Sophia. So let's hope that
maybe Sophia has some reinforcement learning. So in fact, there are 3 different types of learning. For example, there is a perceptron. So it's a kind of simple neural network. Plus and minus. Do you remember this simple artificial neural network? It was created by Rosenblatt in 1953, as I guess. For example, if we want to recognize what animal is in the picture, cat, for example, or not cat, we can show to this simple neural network cats and not cats, and after some iterations, it will learn what is cat and what is not a cat. This is a learning, supervised learning, or learning with a teacher. So when we tell this is right and this is wrong to the network, there is a kind of unsupervised learning. So do you know the examples of unsupervised learning?
we down with this. We're not trying something and after you discover the right experience by yourself.
When it comes naturally, because of the
So in fact, unsupervised learning is about clusterization. No? So have you heard about K-means, for example, no? Some methods of machine learning. K-means, no? No? Okay, so in fact, in deep learning, the structure of deep learning neural networks is something like some kind of clusterization here, and in the end of the neural network, there is a perceptron. So, in fact, the deep learning is combination of unsupervised learning and supervised learning. For better understanding their unsupervised learning, so imagine how our visual system works. When we see different objects, different I don't know, different landscapes, different people, so our visual system learn to detect, for example, some square objects from some round objects, but nobody talk to them. It's a square and this is a round. So it's just because the characteristics of these objects. Okay, so this is a feature of neural networks or clusterization. And the neuroscientist Doya in 1999, yeah, he proposed that all these types of learning, we can find all these types of learning in the brain. So in cortex, in cerebral cortex, we have unsupervised learning, so without a teacher. In cerebellum, we have supervised learning. Yeah, so the cortex is a teacher of cerebellum. And in basal ganglia, we have reinforcement learning. So the basal ganglia is a necessary part for this reward learning. And have you heard about the DeepMind neural network, which plays in Atari games? No? Atari games, yes. So there was a small company, DeepMind, and Google bought this company after their Nature publications on Atari games. So they created a neural network which was able to play some simple computer games. And so they succeeded because of reinforcement learning. So they added reinforcement learning to usual deep learning networks. Maybe I'll talk about this later. Okay, so this is basal ganglia, and they work as kind of filter. I supposed it for myself. And there are different neural circuits with basal ganglia. Some circuits are connected with the motor cortex, some circuits are connected with associative circuits, and some are connected with limbic circuits. Oh, have you a question or something to add? And frankly speaking, I'm not a specialist on basal ganglia, and I've read about them for many years, but I don't understand their function at all. But in some way, so I can tell that, for example, you can memorize about the function of basal ganglia in this way. A normal people with basal ganglia can go from, for example, from his room to the toilet room, yeah, and just walk and do what he or she wants. And if we have got some problems with basal ganglia, we can filter all the stimuli and all the processes in our brain. So, for example, I go to the toilet, and then I see the key, I play with key, and then I go somewhere else, meet someone, and so on. No, I'm not. I remember about my aim, but I can't control. Yeah, I do anything with all the destructors. So I try to do all the processes.
So basically, like filter that.
Yeah, this is a filter. But it's a very simple explanation, but it's good to remember something about the basal ganglia. Okay, and now let's talk about the cortex. And we know something about the cortex, and we knew something about the cortex before we discovered the large-scale brain networks. And these are dorsal and ventral streams. I see this is a dorsal or vision for action, and this is a ventral stream or vision for perception. So this is a visual cortex, yeah, in the occipital lobe, it's here. And maybe you remember, so where is the motor cortex? Motor cortex, yeah. Yeah, here there is a motor cortex between, so it's the posterior part of the frontal lobe. Okay, and we have talked about this fact that all the things which are connected with visual perception, I mean, if you want to find the part of the brain connected with face recognition, for example, So you will find them in the temporal lobe, so somewhere there, inside the temporal lobe. And so the dorsal stream function is the connection of vision and motor cortex. Okay, so that's why we need some special processing for this way. So we don't need very high special resolution. Yeah, it's connected with movement, location, so what is where. And the ventral stream is connected with some special features of the objects, with their color, texture, some shape, size. So we collect the objects somewhere in the temporal lobe. And these are the main characteristics of dorsal and ventral streams. Maybe you will need this table for a test. For example, the speed of ventral system is relatively slow and the dorsal system is relatively fast because it's connected with where, and we don't need any speed to answer what is the object. Okay, and now we are going to networks. So first of all, I want to remind you about the different brain imaging techniques. So do you remember the meaning of this picture?
It's like the injury
What's the question? What you're asking? What's this?
Yeah. What's
It's a different type of brain imaging.
Yeah, there are different spatial and temporal resolution, but we usually use fMRI and sometimes MEG. Yeah, these are different brain imaging techniques, so we can find the activity of the brain with the help of electrode rays, fMRI, mirrors, MEG, EEG, and so on. We know we usually use fMRI to recognize some large-scale brain networks because it's very convenient. So we have rather good spatial resolution, and we can divide one part of the brain from another part of the brain with the help of fMRI. And by the way, large-scale brain networks, they are connected with the function. Okay, so that's why we don't use some static techniques. So we need some functional techniques. Okay. So there are we can divide the networks of the brain on many large-scale brain networks. For example, we can divide them for 100 brain networks, but it's very big number, so we need something lower. And we usually divide the networks on from 5 to 17 networks, somewhere, something like that. Yeah, and they can be presented like here, and or like here, for example. And in 2011, there was publication in Journal of Neurophysiology, the organization of the human cerebral cortex estimated by intrinsic functional connectivity. And this was a quite huge article. And in this article, the authors divided the activity on 7 brain networks: visual, somatomotor, dorsal attention, ventral attention, limbic, frontal, parietal, and default or default mode network. But we will talk about some of them and about some other brain networks. So the default mode network, have you heard something about this network? Nothing? Default.
You're reciting it. You have to remember something that you don't know.
No, I'm trying to go with default as the meaning of default. So
Default? No, that's not connected with economics. Okay, so we also call it default state network, and this network is active when we haven't got any task. So when we just sit and haven't got any task. We haven't got any goal, and we so
Nothing box,
nothing.
like nothing box, like when you are not doing anything and stuff.
Yeah, I don't know how it is in English, but in Russian there is a word пенек. It's a part of the tree, the lower part of the tree, and it
Trunk in English.
maybe, and it means that you sit and do something like that. Yeah, and there are the main parts of the default mode network is precuneus. This is this part, so in the central of the parietal cortex, and also some part of ventromedial prefrontal cortex. Medial prefrontal cortex. This is the simple explanation of default mode networks. So this network is active when you process some information about yourself, about some mind wandering, and it's active when you haven't got some certain task, and yeah, some internal monologue. So this is about default mode network. The next two networks are dorsal and ventral attentional network. So they are connected in some way with dorsal and ventral stream, but also with our
Can I ask a question?
yeah.
In the whole this neuro thing, how can we trust that this is true? Like the brain is just like a big fatty looking something, and you pick some very tiny thing and say, okay, this is responsible for emotion. How can we know that this is true?
You mean for example medial prefrontal cortex or the whole network?
When you dedicate just one tiny function to it and say, okay, this is the center of the activity, how would you know? Like sometimes I ask myself a question like, how do we trust that this is true?
Oh, the answer is quite simple. So first of all, you should do a lot of experiments.
Yeah.
So I mean, all the neuroscientists do some experiments. For example, some people have lesions. Okay, so for example, this guy had got a medial prefrontal cortex, and he
The ability to do so.
he had the ability, hasn't got the ability to do something. Or for example, we can use transcranial magnetic stimulation when you off some parts of the brain, but you can't off some limbic system or so on.
Breakdowns is a huge experiment, and also that breakdowns to filter during the filter experiment to reach the specific part.
Yeah, you can also you can do some lesions for rats and for mouses, and you can use drugs to off some parts of the brain. You can use optogenetics to on some parts of the brain. Yes, and also, so you need some logic to explain these functions. In some experiments, when we ask people to do something, for example, solve the problems, and we scan the activity of the brain using the fMRI, for example, we have 100 people who solved the problem in a good way and 100 people who solved the problem in a bad way. Then we analyze what's the difference between activity in functional in fMRI, and we find what's the difference. So the activity of what parts of the brain are positively correlated with good solutions and negatively correlated with good solutions, and so on. So we can't tell that some special part of the brain or some special neurons, only them are responsible for something. No, we don't know that. It's just a combination of different facts and different experiments. Okay, so the dorsal and ventral attention network, they also can be combined in oriental attention network. So we discussed it last time here. The visual network is very simple. It's a visual cortex. So V1, V2, V3, V4, and so on. There is one more interesting network. It's a salience network. Its function is not quite clear, but it's connected with salience. So when we want to know what is more important, for example, in the room, the salience network helps us to do that, to find out which things are more important.
In terms of what, like priority?
In terms of the brain.
It depends on the aim of room.
No, I think it depends on the person.
Yeah,
you will neglect this and pick this.
No, it depends on the room. It is a classroom, so I think, in my opinion, it's important to project of the whiteboard.
Yeah, but it's a certain mode.
Yeah, so it is individual.
Yeah, of course, it's individual. But if you want to find out what's more important, you need to use your salience network. So the main parts of the salience network is anterior singular cortex and insula. So anterior singular cortex is a very interesting part of the brain, and it's widely used for some other explanations and experiments, because, for example, it's also active when you solve some examples like that. If you solve this problem, your anterior singular cortex is active. Or, for example, if you find out some errors, this part of the brain also is active. Or
this part doesn't work.
No, if you want to compare something, this part is active, and so on. So it's necessary to compare, to find errors, and so on. And for example, the activity of this part is different during the hypnosis, and we will talk about this later.
Inactive or active?
It's not the same, and it depends on the person. Okay, and one more neural network is frontoparietal network or central executive control network, and we need this network to achieve our goals. So to do some tasks with our working memory, and there is a model of there is a triple neural network model. It looks like here. So default mode network, central executive network, and Selens network. So there is a theory that the Selens network is intermediate network for default mode and central executive network. So usually we have two modes when we have a certain goal and when we haven't got any goal. And when we switch between these modes, we are in a Selens network. Okay.
In between them?
I mean, for example, now I'm a default mode network. I'm wondering, I don't have any mode. Then my brain is switched on Selens network, and I decide, I need something to eat. And then I have a goal. I need some food. And with this goal, I go to the wedding automat or to the cafe, and then my central executive network is active. Okay, so we have this theory because the activity of these networks are anti-correlated. So they're not active at the same moment. Yeah, very interesting. And the main parts of the main hubs of the central executive networks are dorsolateral prefrontal cortex and posterior parietal cortex. Okay, and these are these networks again, the triple network model. Again, with some explanations of how does it work. So we can divide the networks in different ways. For example, we can also divide it on auditory cortex, because we know the parts of the brain which are connected with auditory things, sensor motor, and language, for example.
I have a question.
Yeah.
Generally, for people who lack a sense, like hearing or visual sense, it's say they are more intelligent. So it's somehow related to this, like some of those network combined to another. Or
usually people, for example, can see something because of they have got some problems with eye, and in this way they can use the visual cortex for something else. And if they have got problem with the visual cortex, they can't use it for anything else. So it depends on the type of the problem. Okay, and so have we talked about the connectivity? No. I mean functional connectivity, structural connectivity, and all this stuff. Okay, just a short idea about the connectivity. so there are different types of connectivity. The structural connectivity means that we have a connection between two brain parts, so physical connection with neural axons. Yeah. Functional connectivity means that these parts of the brain are active at the same moment, and so effective connectivity is connectivity when we know which part of the brain affect on which part of the brain. Okay, so we know the direction. And so what do you think about all these large-scale brain networks like salience network or executive control networks? Which kind of connectivity is it about? Yeah, that's right. It's about functional connectivity. So we don't know if there is a physical connection between two brain parts, but we know that these parts of the brain are connected at the same time, and they're connected with some function, like achieving the goal, for example, for executive control network. And also we can find out some brain networks with the help of EEG, or with the help of some computational methods. For example, if we have got the continuous activity of the brain, we can do some kind of clusterization or find out some principal or independent components, for example, like here. So there is a special software for fMRI, for example, and you can find out different components of the activity in the brain. And in this way, we can tell that, for example, this is a component with this, which is connected with some brain network, and this is some noisy component. So it's the way how can we find out these large-scale networks. So we use some kind of clusterization of the whole activity in the brain. I think we should finalize our lesson, but before that, I just want to remind you that some networks in the brain, they are already in the artificial intelligence, and for example, the convolutional neural networks, they are inspired by our visual system. Yeah, so like if the first layer is connected with retina, and then LGN or lateral geniculate nucleus, and then V1, V2, V3, and so on. Okay.